So I think we'll get started now. So good day, everyone. Happy second day of summer. Hope everyone's off to a good start. Today, we're happy to have Mowei Zhou with us from PNNL to talk about some of his recent work on plant proteomes. I know a lot of us work in model organisms and human systems, so it'd be really interesting to hear more about some of the other things in the universe. So let me just give a couple quick little slides on some of the activity of the consortium. One of the main things we like to do is to thank our sponsors who enable this. Thermo Fisher, Brooker, and SciEx been very generous, as well as our supporters, Agile and Pfizer, allow us to do these types of programs and continue to grow the community and address some of the challenges that we all have out there. Just one thing I want to mention is some of the resources we have at our website. I mean, some of these are a couple layers down, so you may not see them at first glance. One is we have a variety of methods that may be helpful for people, both if you're looking for an on-ramp to some of the top-down approaches, or you have some challenges. We have a few up there. One is around a protein separation process, and we have two methods for Brooker instruments and thermal instruments. They have standard information as well as details and instrument settings, if you want to take a look at those. Then we have some information on antibody digestion. Just an example on the protein separation methods. We have this paper. There's about 10 specific methods or so, protocols in here. And based upon the type of sample you have and the kind of information you want, there's a sort of a flow chart that helps you get through what's the best approach as far as sample preparation, sample cleanup, and separation. If you're looking at native things that have certain detergents in them, that's a lot different than if you're looking at simple MS-compatible buffers and denatured samples. So you can take a look at that paper. There's a lot of really good information there. It's been validated across different laboratories, so that these are more or less robust methods that you could go ahead and start with and optimize for your own systems. There's also a list of the publicly available software. If you happen to have another software tool that's available for the community and you'd like us to mention that as well, please send me a note and we can go ahead and include that in the list up here. We don't really have any guidance around these. They all have their own set of help files and GitHubs and everything. So you can take a look at these and see if some of these would be helpful for you. We know the software is a big challenge for everyone, and we're trying to figure out ways that we can help people with that as well. But that's a big challenge, we know. So anyway, here's some resources for you to take a look at. So if you're not a member yet, you can go ahead and join the community. It's free to join currently, and you'll be first to know about new programs, projects, things like that. So please take a look there, and it'd be great to have you on board. So let me hand it over now to Mowei Zhou. Is in the Environmental Molecular Sciences Division at PNNL, and she'll be speaking to us about understanding the heterogeneity of plant proteoforms by top-down mass spectrometry. So go ahead, Mowei, it's all yours.
Thank you, Paul. Let me share my screen. Paul, could you unshare your screen? So I think I can just overload it. Okay. Okay, so thanks, Paul, for the introduction. And I've been attending this webinar series. I've really enjoyed it. So I'm really honored to be able to give a talk here to talk about some of my recent work on plant proteoforms. And as Paul mentioned, we've seen a lot of work in human and some other systems of some other model systems, but there's a lot of interesting unknowns in the plant research. So I hope I can convince you top-down can really play an important role in plant research as well. So, no. Okay, sorry, it was a little slow. So I think most of us are very familiar with the topic about top-down. The reason we're trying to develop top-down techniques is really advantageous to study proteoforms. And as most of us know, from a single gene, there are a lot of post-transcriptional and post-translational modifications that give rise to multiple proteoforms for the same gene.
Mowei, excuse me, we're not seeing your slides for some reason. If you could just kind of reshare, maybe.
Okay. Somehow it was unshared.
There we go. That's good.
Okay.
Great. Thank you.
Yeah, sorry about that. It has been very well documented that the different proteoforms are very important in human diseases. So a lot of common post-translational modifications like phosphorylation, glycosylation, they all make the proteins into different proteoforms, and those proteoforms are directly correlated with different disease states. And as a top-down community, we know that there are different advantages and disadvantages for different techniques. And the bottom-up proteomics is very widely applied for detecting proteins. It relies on enzymatic digestion of the intact proteins into smaller pieces of peptides, which are easier to detect by mass spectrometry. So the proteins are inferred based on the detection of those peptides, or maybe subsets of peptides. It's a very robust and highly sensitive method, but there are some challenges of getting complete information of the proteins. For top-down, we bypass the digestion steps, so we directly look at intact proteins. And I really like this picture from Northwestern to kind of really give you a high-level idea of the difference and the benefit of combining both top-down and bottom-up. Because with the top-down, you really get the high-level view of the proteins. We can really be able to better piece up the smaller pieces together if we combine the top-down and bottom-up analysis. Although currently we know that the top-down is more challenging to perform than bottom-up, but I think with the new developments in the field, we're really hopeful that the top-down will be more common practice with efforts from the community. So why we want to study plants? This is actually a very easy to understand question, because a lot of important molecules are actually made by plants or made with plants. So this is a nice quick overview of a lot of plant materials, like the natural products from plants. They are very important for a lot of the biological and the ecological processes. And as humans, we know we consume a lot of plant materials, either as food or cosmetics or even medicine. And there are also some interesting effects of some plant products, like the catnip can really make cats happy. So in addition to those, there is also a growing interest in renewable energy because of the climate change challenges. And there's a lot of efforts under Department of Energy to develop new ways to better utilize biomass for renewable energy. And basically, you can think about using those plant materials and convert them into fuels, so that those are better for in the environment. And plant proteins actually regulate a lot of those processes to produce those molecules we want. And even if you don't think about those molecules, if you're into plant-based diet, that is known to be better at reducing greenhouse gas emission because of the production process is more efficient, and those are actually plant proteins we're eating to achieve this goal. So there are a lot of important materials and molecules that are made from plants we want to better understand, especially for plant proteins that can regulate the production of those materials. So if we can understand how they work, we can better engineer solutions to have more efficient production of those molecules. So this is just an overview of some of the plant-related research I've been working on in recent years. And some of them are already published, so I won't talk about in detail, including some histone post-translational modifications in the sorghum plant that were grown in the field under the drought conditions. So we're trying to find epigenetic markers that are related to drought response of the plants to develop more resilient plants for the harsh environment. There's also an interesting study recently published to study heteroprotein complexes in plants. And I'm not going to talk about details today, but you're welcome to look at this paper. What I'm going to focus on today are those two projects. One of them is to study soybean root nodule proteoforms, and we found some unexpected truncations and also modifications. This is still unpublished data. And the other one is also a recently published data, but I also want to talk about something that was not highlighted in the publication, where we're trying to purify some proteins from plant stems and then try to sequence them without having the full genome of the plant. So I'll start with the soybean nodule project as the first example. So the soybean nodule are actually involved in the symbiosis. So for soybean, if you look at the root, there are those soybean nodules which are actually bacterial infections. So this bacteria is very important for nitrogen fixation for the plants to give the plant better nutrient. And this process is very complicated. There are a lot of studies in the field to try to understand the whole process. So if you imagine this process, you actually have the plant allowing specific bacteria to go into its root, and then they will live as like a symbiotic relationship. So there's a very fine balance of symbiosis versus immunity, because the plant cannot just let every microbe in, because some of them can be pathogenic and harmful to the plants. Most of the studies so far have been focused on metabolomics and some of the bottom-up proteomics, but in this case, we'll be showing some of the top-down analysis. To give you a better view of the structure of the soybean nodule, this is a cartoon figure of the actual soybean nodule. So you have the plant root, and then in this nodule, in the middle of the nodule, this is the symbiotic region where the bacteria is localized. So you would expect to see most of the bacterial proteins inside this symbiotic region, while the outside will be just the plant materials. So for just a little bit background on these samples, so this is in close collaboration with Professor Gary Stacy at the University of Missouri, who has been very generous in providing us very great samples for those soybean nodule studies. And a lot of my colleagues have been working with him as well in the past few years. I know there are several publications focusing on metabolite imaging. And recently, we've done some plant glycan imaging and shotgun bottom-up proteomics. So this shows that the representative optical image of the soybean nodule slices, tissue slices. So the soybean nodule is inoculated by the Rhizobium japonicum bacteria, and Gary's group has really optimized those two strains. One is the Y type, the WT, the Y type, and also the other bacteria was a mutant that lacks one of the gene for one of the nitrogen fixing proteins. So this mutant cannot fix nitrogen, but it will still inoculate and infect the soybean. So it's a very nice model system to study this symbiosis process, because in the Y type it actually does fix nitrogen, so the whole process is happening. But for the mutant, you only infect the soybean, but you don't fix nitrogen. And most of the previous studies were doing the multi-imaging, so matrix-assisted laser desorption ionization to study the small molecules. And there has been some mechanistic studies in those processes. And for the study I'm going to show, we're really trying to see what kind of proteoforms we can detect in this soybean nodule. And there are some significant differences between the Y type and also the mutant. So this is an overview of the volcano plot to summarize the quantitative top-down proteomics results. So I really want to thank James Faucher, who has been instrumental to do all those data analysis in a very automated manner with his topic R package. So we can easily do some clustering to quantify the proteoforms among the samples. So for this study, we have 5 biological replicates for the Y type and also the mutant. And also Dushan, Maria, so they were really helpful in preparing the samples, and Stephanie and Yan Chen was helping with the shotgun proteomics analysis. So in this plot, we have the higher abundance proteoforms in the Y type on the left, and the higher abundance proteoforms for the mutant on the right. So we separated the plant proteins from the bacterial proteins and normalized them separately, because the ratio of the protein seems to be changing a little bit between the Y type and mutant. So this is a more fair comparison for the change within the plant proteoform and also within the bacterial proteoforms. And we were pretty happy to see some expected changes. For example, this NFH fragment. So we did, this protein is quite big, but we were able to detect a small fragment from this protein as one of the proteoform, mostly in the wild type sample, because this gene was knocked out. So we hardly detected anything in the mutant. So this means that we're performing this quantitative proteomics, top-down proteomics correctly. Oops. The other thing I want to mention is that we see a lot of proteoforms from this like hemoglobin, which almost consisted about 25% of all the quantifiable proteoforms. So this is a very, very highly abundant proteoform in this sample, which is also expected. And a little bit more on this like hemoglobin. So like hemoglobin, it regulates oxygen level in the root nodules. So it's very important for the symbiosis process, because the bacteria, when they fix the nitrogen, those nitrogenases are very sensitive to oxygen. So you have to really control the level and keep the environment as a like anaerobic condition. But also the plant, the metabolism requires oxygen. So the neck hemoglobin really regulates the oxygen level in a fine-tuned manner so that it keeps the process going. And for the leg hemoglobin, there's an equivalent protein. Basically, the hemo is very similar to the mammals, to the mammal hemoglobin protein. It binds heme. So if you look at the actual tissue, you see this red color. That's actually come from the leg hemoglobin because of the iron in the heme. And that's also the protein that was in those plant-based meat to give the actual feel of a real meat. And I even Googled, it was interesting that the companies were actually using yeast to recombinantly express neck hemoglobin of the soy, and then add that into the meat to make it really taste like a real meat. But what's interesting is that when we look at just the leg hemoglobin proteoforms, we see a very drastic difference regarding the truncation of leg hemoglobin. So the dots are the different proteoforms, and the full-length leg hemoglobin are the purple dots, and the yellow dots are the truncated leg hemoglobin. So again, the higher abundant proteoforms for the wild type are on the left and the mutant on the right. So we saw a lot of full-length leg hemoglobin in the mutant, but in the wild type, we see a lot of truncation events happening, which is quite interesting. And then James also had the script to plot this coverage map. So the x-axis basically is the protein sequence from N-terminus to C-terminus. And those bars are basically detected proteoforms to show which fragment we're detecting for the specific proteoforms. And we detect a lot of them, like a lot of the smaller fragments, and the color represents the relative abundance. The color represents, the spectral matches, the spectral counts for each of the proteoform. So we're mostly detecting the intact proteoforms, but there are some specific fragments that seems to be also very highly abundant. And if we look at a pattern, it looks like there are also some specific cleavage sites for those truncated proteoforms. So we're still looking into some other data like the bottom-up proteomics, and maybe even looking into some metabolite data and see if there's any correlation or clues to help us understand why we're seeing a lot more truncated proteoforms in the wild type. So it could be that there's just a higher turnover rate because the wild type is really actively doing this symbiosis process. So maybe there's a higher turnover rate in the wild type, or maybe there's some like regulated protein degradation. I don't know if there may be some bacterial proteases that are trying to make sure the hemoglobin doesn't bring too much oxygen in the bacteria in some symbiotic region. So this open question we're still investigating and trying to get more information from other data. But this really shows us how the top-down can pick up those differences. And if you imagine if you're doing bottom-up, a standard bottom-up analysis, this kind of cleavage events will be very difficult to extract. Those kind of information will be likely lost. And I don't have time to show the bottom-up data, but when we were doing the protein quantification, we basically see the like hemoglobin doesn't change much. The total protein abundance doesn't really change much between the wild type and the mutant. So this highlights the benefit of doing top-down analysis to study the protein truncation events. So the next topic I want to mention in the soybean nodule paper, soybean nodule project is the ability to really discover unknown or unexpected PTMs. So there are a lot of software tools that can do the open search tool, and I usually use this topic suite from Xianwen News Group. And the way I do this is like when we look at the results, we just sort the proteoforms in the Excel table. And in this software, when you have unexpected modification, we'll see a mass shift annotated on the proteoform. And in this case, I'm looking at the one specific protein, so all the proteoforms for that specific protein. And when I sort this table, I saw a lot of proteoforms with very familiar mass shift. So you see mostly 317 or like 341. There's several others, but you do see a high frequency of 317 mass shift at the N-terminus. So just to let you know, we published a book chapter in this proteoform identification edited by Professor Liang Sun and Xiaowen Liu. It described more detail on the steps we typically use to identify the unknown proteoforms and validate that manually using different software tools. And I won't talk about details here, but I just want to give you how we kind of apply this approach to confirm or identify what this unknown mass shift is. So to better display the unknown PTMs in the search results, we plotted this histogram of the mass shift and also plotted the unknown mass shift against the spectral counts, the PRSM, so the proteoform spectral matches. And this gives us a very easy way to visualize the frequency of the different modifications. And we'll typically focus on the proteoforms that the unknown modifications that have a high frequency, just in case that some of the single hits may be a random or like false positive identification. And a lot of them are like -1 or +1. So those could be the isotoping errors, which are very common in top-down results. So, but some of them are actually disulfide bond, which I'll show some examples later. And also another common one is oxidation. Some of them can be from artifacts. We're not discussing that here. I do want to highlight there is this cyano modification, which means that we have cyanylation on 15 molecules. And we have very good data to confirm that. I'm not going to discuss that in detail here, but we can chat if anyone is interested. And also we see some 80 Dalton mass shift. Some of them could be phosphorylation, but we also have some spectral suggesting that's actually sulfonation on the serine, serine, or tyrosine residues. And we also see some cystination, which is also interesting, some glutathione, and also this 317 Dalton mass shift, which I'll focus on in the next few slides. Also, just to want to mention that we have done this analysis in the past for some other plant, not plant, but this is cyanobacteria proteins. So there are some photosynthetic proteins in cyanobacteria where there is this chromophore covalently linked to the protein. And if we do the same analysis of the histogram, we can actually look at how the chromophores can be partially degraded on the protein with the same analysis. And you are welcome to look up this paper if you are more interested in this kind of PTM analysis. So to give you a more detail of how we look at the data, so for this 317 Dalton mass shift identified by this top-down search, we saw this modification on actually 2 homologous proteins. So the accession numbers are here, but when we look up the UniProt database, they're annotated as uncharacterized protein. And all the homologous proteins are also annotated as uncharacterized. So that's great. We really don't know what these proteins are. So there's not much information we can rely on to figure out what these PTMs may be. And this is the sequence alignment. So those 2 proteins are very homologous, but they really have the same identical sequence at the N-terminus and where the modification is. And AlphaFold also predict them to be both highly disordered. So it's very hard to figure out what they are. And when James figured out this out, he was so excited and was telling me, I figured this out what this is. So he was saying like, there's actually, this methionine is actually not there, which is totally expected. So there is a 210 Dalton mass shift here. and then there's a 238 Dalton shift here. And now we can actually match these masses. So the first one is the meristylation. It's a lipid modification at the N terminus. The other one is the palmitylation, which adds on the cysteine residue. So to just convince you that we're really detecting this, this is the top-down data. This was visualized in LCMS Spectator developed at PNNL. So this shows the annotated MSMS data for this proteoform, and then it also matches the MS1 precursor peaks really well. So we're very confident that this is matching really well. And then this is the sequence coverage map, which we had about 62% coverage, which is not bad at all. And to further convince you that we are seeing those two modifications, we do have this Y ion here to help localize the palmitylation at the cysteine. And then the rest of the mass shift goes to the N terminus. We're a little still suspicious on this kind of detection, so we also looked up the bottom-up data, the matching bottom-up data of the same sample. We look at this results, and we found this N-terminus peptide here, but we were doing this shotgun bottom-up with DTT, the cysteine reduction and also alkylation. So we were not able to confirm the cysteine palmitylation directly. We're just seeing the alkylation product. But we were able to identify this N-terminal meristylation to complement our identification in the top-down. So when we look up the literature, so James found this paper over 20 years ago saying that this is actually not unexpected. The meristylation is known to help anchor proteins into membranes. But with just this modification, the anchoring is not that stable. So this is kind of a dynamic process. To stabilize this interaction of two membranes, there's this known two-signal model. So you can have other additional factors to stabilize this interaction. So one of them is this meristylation plus palmitylation. So if you have two lipid molecules added to the protein, it will significantly stabilize this interaction and help the protein to stay close to the membrane. So there are also other papers I'm not going to discuss in detail here, but there is some predicted sequence motifs for this kind of modification. So you have this meristylation, and then when you have this N-terminal meristylation, if you have a cysteine residue nearby within like maybe 4 or 5 residues, you can have this double lipid modification. And our experiment really confirmed this PTM from the predicted motifs. And I also want to argue that if you are doing bottom-up proteomics, this peptide with 2 lipids on a very short peptide may be difficult to detect. There has been some work on studying palmitylation with bottom-up proteomics, and typically you require some special protocols to better detect those hydrophobic peptides, or even doing some chemical substitution to make them easier to detect. But with top-down, we can easily see this because we are still detecting full proteins, which helps them solubilize and kind of even out the hydrophobicity of the PTMs. So in addition to the LCMS, we also attempted to do some multi-imaging to study the spatial distribution of those proteoforms. So this is just, if you are not familiar with the multi, so we have this laser shooting on the tissue section, which are coated with matrix. And so we raster the laser across the tissue with small steps. In this case, we were doing 15 microns, and then we can actually get the mass spectra and then the images of those proteoforms. So most of those have been done with metabolites, but Kevin has been really great in developing this for the high mass proteins on our UHMR proteoform. I forgot to put his paper preprint here, but if you're interested, I can give you the link for the paper. But we use that UHMR high mass system with the multi front end to map those proteoform spatial distributions. So just briefly, you can see that this is the optical image of the soybean nodule. This is the mutant sample. So the inside is the symbiotic region, and the outside is the cortex. It's more like the plant part. And then you can see there are a lot of peaks in there, and then many of them have distinct spatial distributions. They're either in the symbiotic region, or they're in the cortex region, or they may be in both regions. and some of them are only in the symbiosis regions. So what we were trying to do is to assign those peaks based on the accurate mass by looking up the proteoform identifications from our previous LC-MS results. So it was actually quite challenging for the sample because we do see quite a lot of peaks that we couldn't really assign very easily. So those peaks, you see a lot of peaks, they're actually spaced by like 14 Dalton, so it looks like some kind of algae chain. And we don't think it's from contamination. It may be some endogenous plant polymers that are about 3,000 Dalton. And then we didn't assign this one, didn't assign this one. So this one was assigned as a cold shock protein from the bacteria, and it seems to be localized in the symbiotic region, which makes total sense. What's interesting is that we see some plant peptides and bacterial peptides that seem to be having same spatial distribution. So overall, this experiment was a little challenging because there's a limited overlap with the LC-MS data, maybe because of the different detection biases that needs to be further optimized. But we saw some interesting peptides, as I was mentioning on the previous slide. So just to give you an example, this is one of the bacterial peptide we identified in top-down LC-MS data, and we matched the accurate mass in the multi-image. And you see that we detect some disulfide bonding in this peptide. So the structure is reasonably good. So we think this is a real identification. And then we also see the plant peptide again. This was also identified with cysteine residues in here that are likely to be in some kind of disulfide form because we don't really see much sequence coverage in this region. So we think there may be some cross disulfide bridges that are protecting this from fragmentation. So both of them are disulfide peptides from a big protein sequence. This is actually not that irrelevant biologically because there are some known secreted peptides. Many of them are also known to have disulfide bonding to just stabilize the structure. Those tend to be some signaling molecules for recognition. So we think maybe those are actually not just some random degraded proteoforms from artifacts, but maybe some of them are specific to some signaling pathways. And this is just a very broad hypothesis. We haven't been able to look further into that, but I think this shows how this kind of combined proteoform analysis can really help us to discover those potential targets for further biological studies. And again, I want to highlight that all those proteins, if you look up the UniProt database, they are annotated as uncharacterized protein. So we really don't know much about them. And this really gives us some clue to start to understand the potential biological function of those proteins. So just a quick summary on the soybean part. I know I'm kind of maybe running a little bit short on time. So I hope with this data, I convince you that the top-down can be really useful for this hypothesis-free proteoform discovery, especially if you do this open search analysis. And it can really help us understand those uncharacterized proteins by studying their PTMs and also spatial distribution when we combine that with imaging techniques. And this integrated analysis can be really helpful to understand the heterogeneity, both regarding the PTMs, the truncations, and also the spatial distribution. Especially for this kind of interspecies communication, there may be a lot of signaling molecules that are secreted that are very useful to study with top-down. The challenge is that the detection biases among different techniques may make it difficult to integrate the data. But I think that will slowly get resolved as the protocols are further optimized. So I want to maybe quickly go over the next project that was already published, but I want to kind of highlight some of the components that we didn't really highlight in the paper. So the idea of this trying to understand in unknown proteins is actually very useful, especially in the understudied organisms, like some plants or some microbes. Well, if you look into the databases, a lot of them are poorly annotated, and there are a lot of protein dark matters in those organisms. And for plants, especially, a lot of them have very big genomes. So this is the genome size for different species in base pairs. And the mammals are here. They have very narrow distribution. But you see the flowering plants, they have a wide range of genome sizes, and a lot of them are spanning into very high, the highest, the largest genome you can see across different living organisms. So the big size makes them difficult to sequence and annotate, sometimes just purely because of the size of the work you have to put in. And if it's not a very popular organism for funding agencies, it's hard to actually get even funding to annotate them. And there's a recent PNAS paper that estimated about only 0.2% of known species in the green plant clade have been sequenced. So there's a lot of unknowns just because of the challenges to sequence those genomes. But if you imagine across human history, a lot of plants are actually used by humans, like a lot of natural products are known to have activity, and even some biochemistry activities have been measured in modern science, but without having a full genome of the protein, of the organism, which really limits what we can analyze regarding the proteome. So this was actually one of the projects I got involved with Professor Norman Lewis and also Dr. Lawrence Darwin at the Washington State University at Pullman. So this is basically the case where they published over 20 years ago. They studied this Forsythia intermediate plant just because they have a lot of material they can collect from people's gardens, or it's like very easy access material. So they were able to purify proteins with a specific activity they were looking for directly from the stems. And then they identified this family of proteins and they named dirigent proteins, which this whole family mediates stereo selective coupling of phenolic compounds. And in this specific protein, dirigent protein, this DP they identified, it can actually mediate the coupling of the E-coniferol alcohol to this plus penoresinol in a very stereoselective manner. So the hypothesis is that the plant will bind those radicals once this is oxidized and force them into a very specific orientation to give this very specific stereochemistry. Without this enzyme, if you just oxidize this compound, and you will form all those racemic products because you don't have control on the product stoichiometry. So what's the significance of these dirigent proteins? So there are two parts to this story. So one is there's some potential for bioenergy applications, because there was a paper from another group describing how they localized a homologue of the dirigent protein in Arabidopsis root. So it was localized in a very specific wood structure in the root. So it was hypothesized that this protein is somehow involved in this lignification process. So if you imagine, if we can really understand the mechanism, we can probably engineer plants that have different properties of the wood, and possibly making the wood structure easier to degrade for bioenergy applications. And another more recent study, which is also very interesting, is that they use this dirigent protein as a catalyst to perform this very selective selective synthesis of a natural product analogs, which are used as anti-cancer drugs. So those syntheses are very difficult to do with traditional chemistry. But if you can imagine using those enzymes, it will be a more efficient way to produce those difficult to synthesize analogs of natural products for biomedical applications. So that's kind of the motivation. So why we want to study more of those dirigent proteins? I just mentioned like a few of the homologs in the previous few slides, because the dirigent proteins is a large family of proteins. So this is the phylogenetic tree of dirigent protein homologs in sequenced genomes of plants. So each line is actually one homolog. So you see there are a lot of them. And only a few of them have some level of characterization, either biochemical or like a phenotypical characterization. And even for just a single plant, Arabidopsis has 25 homologs. And some plants can have even 40 to 50 homologs, and we don't really know what are their functions. So that's why we really want to study this. And I want to go a little quicker just to show you like when we started the project, because we really don't have the genome of the Forsythia. So we're trying to see, can we actually do some de novo sequencing to figure out what proteins we're purifying from the plants. So we used all kinds of mass spectrometry methods we can think of. So the de novo peptide sequencing gives us sequence tags of the peptides, and we were able to get some transcriptomics data from a similar plant, not exactly the same plant. And then we combine them to create, and also the top-down data, we have some top-down data to combine with those bottom-up and transcriptomics data to create those candidate sequences and validate them. And then we also perform some native mass spec to study if there's any complex formation among those proteins. So a little bit more detail on how we actually did this in this project. So this was really, I also got a lot of help from Cannon Bell, who helped assemble those transcriptomics data and also the de novo sequencing triptych peptide data to give me a candidate protein FASTA database. And for the top-down side, the native mass spectrometry data helped us target what masses we're going after just by looking at what complexes or proteins we detect. And with the top-down, we can get manually actually do some de novo sequencing to get sequence tags for some of the masses we're going after. So we can look up those sequence tags in the custom protein database and further manually validate those sequences. And then once we confirm this, we can reanalyze the data to confirm it. And there was a pleasant surprise towards the end of the project, which is a publication of Forsythia suspensa genome published. Again, this is not the exact plant we're studying. We're studying this Forsythia intermediate plant. And then the suspensa genome, sequence genome, is a parent plant of this hybrid. So we don't have the full genome, we have like 50% of the genome, but that really helped us validate we are getting the sequences correct in our de novo analysis. So I will be quick on this. So there was a serendipitous discovery of 2 new different homologs we didn't expect. And this is just the intact mass in the denaturing of CMS. We see those 2 proteins, and they have different sequences from what's previously known. And one of them have 2 N-glycans, the other have 3 N-glycans. And this protein, the different protein family, they form homotrimers. That's kind of the conserved fold of the protein. And those 2 new homologs are different from the original pano-resonal forming different proteins. And they have distinct sequences to be classified to new function instead of the same pano-resonal forming activity. So we still don't know the activity yet. What's interesting is that when we're doing the native mass spectrometry analysis, we detect the heteroprotein complexes between the 2 new different protein homologs. So you don't have to read the data in detail, but this just shows the native mass spec intact mass data of the different peaks, and we can assign that to different stoichiometry of trimers for those 2 different protein homologs. And we can confirm the stoichiometry by isolating one of them and then break it apart with a collision-induced dissociation. And then if we do this experiment for different peaks, we can easily see that the ratio of the 2 different monomers will change depending on which stoichiometry we started from. So this really confirms the assignment of the stoichiometry, the stoichiometric intact mass. So this is kind of the summary highlight in the published paper. You're welcome to read more details. We also did some molecular dynamic simulations of Arabidopsis different protein homologs to show that this heterocomplex formation may be more universal than just the one we detected in the experiment. We still don't know what's the function of those hetero association, but it is known that some of the heterocomplexes formed between protein homologs, for example, some pseudoenzymes, they are known to fine-tune the protein function because you have basically 2 protein homologs with slightly different function, or even sometimes very different functions. If you form the heterocomplex, the complex will have a tunable function depending on how you mix them. So I won't have time to discuss more details here, but you're welcome to look at this paper, which is, I mentioned at the beginning, this is more better characterized system where we have those heterocomplex formation to tune the activity of this enzyme. So there may be something interesting behind this heterocomplex formation we still don't fully understand. And the mass spectrometry methods really help us discover those. So before I end my talk, I just want to have some discussions to maybe stimulate some discussion around the topic about de novo sequencing from top down. So I just want to reflect on what I did. I was a little bit ambitious at the beginning, trying to do de novo sequencing from top down. So it's actually doable to some extent. You can deconvolute your spectrum or even not deconvolute it. It's easier if you discharge them. And then you can find those sequence tags very easily. And this is not the first time people are doing. I know quite a lot of people even in the audience have done that before. And you can find those sequence tags to sequence part of the protein. The challenge is that even though I tried multiple dissociation methods with the electron-based methods, the electron transfer dissociation and the collision-induced dissociation, once you have PTMs like glycosylation, that really complicates stuff to and limits your sequence coverage. So with the limited sequence coverage, you can really not go very far. That makes it hard if you don't really have a genome or some kind of reference sequence to compare with. But once we integrated the bottom-up data, the transcript data with some candidate sequences, and they don't have to be perfect, it just have to have some template to be able to fit to. And then you will have some information to be able to find the sequences. And I will still say the top down is very important. I don't have time to show too much detail, but at the proteoform level, you get that data from the top down. So you can easily confirm like terminal residues or like a post-translational modification. Like if you have multiple glycans, you can really determine the stoichiometry of those PTMs and validate your sequencing result. So can we go one step further to do native de novo top-down? So it's something I was trying with limited success. So I will share some of the results that was in the supporting information. It's hidden there in the paper, but not really discussed much in detail. So there was a interesting protein complex with very clean signal in the native mass spectrometry data for the protein extract from the plant, Forsythia plant. So this was collected on this water synapse G2SI. We can see this protein complex 139 kilodalton. We can do complex up experiment to dissociate the monomers. And we see there's a 23 kilodalton monomer. So we know that it is a homohexamer. And then the monomer also binds something that looks like a 53 dalton mass shift. It's likely a non-covalent binding based on some other information. So this experiment tells us the stoichiometry of the complex and the potential binding of some ligand or maybe metal. And can we actually do native top-down to figure out what this is without having too much other information? So this was also hidden in the paper, this supporting information. So I did this native top-down with USMR, and it just happened that my colleague Jed Shore had this 157 nanometer UVPD there. So I was doing this 157 nanometer UVPD on this protein complex. I was able to detect the fragments, but only a few of them. You can see I was only able to sequence a few of them manually. I can get a very small sequence tag here. And then by using this sequence tag, I looked up the candidate sequences. I actually found sequence that's blasted to germline-like protein. And why I think this is a germline-like protein, because this germline family protein is known to form hexamers and also bind manganese, which will explain the mass shift of 53 dalton in the previous slide. So just with very little information, but you get different pieces from different places, you put them together, you start to have some information on what this protein might be. To validate this, we also did a cell-free synthesis of this homolog detected from the plant. And this was the cell-free was done by Irina, my colleague Irina, who did the cell-free. Unfortunately, this detected homolog was not folding very well. So we see the proteins trying to fold. This is native mass spec data of the cell-free recombinant, cell-free expressed germline protein. We see it's trying to assemble into large oligomers, but it didn't go all the way to the hexamer. So maybe we didn't have enough sequence coverage, maybe some wrong segments, or maybe it's missing some critical PTMs. But just to say that the cell-free works well, because we also found another homolog from bottom-up data, which has a lot higher coverage, sequence coverage, so we're more confident in sequence. When we do cell-free of this one, it actually folded to the hexamer, mostly to the hexamer. So that at least means Our cell-free works fine. It's just maybe we're not getting close enough with the native top-down. But I think we're getting roughly right about this protein being a germline protein. But it's still needed to have higher sequence coverage to be really able to capture everything. And especially if we have unknown PDMs, that will be even more challenging. And some of the PDMs may be essential to assemble the protein complexes, which I don't have time to discuss in detail right now, but I'm happy to talk about that in more detail. To wrap up, I just want to summarize that the top-down mass spectrometry is a very valuable tool for discovery of proteoforms. And the proteoform can have a lot of different forms, including the chemical modifications, terminal truncations by proteases, and also the higher order structure, for example, the heterocomplexes between homologs that can have potentially interesting functions. And the integration of multiple techniques is very, very beneficial because a lot of times you cannot just solve the problem with just one technique, especially for very challenging biological systems. And also at the end, I would hope to convince you that there are a lot of unknowns that are worth exploring in plants and maybe some other understudied organisms, where the mass spec can really contribute for this kind of discovery science. And especially like spatial omics to study the spatial distribution, and also genome sequencing if you don't really have a full genome available for the conventional proteomics studies. And I want to end with the acknowledgement. I did highlight some of the people who helped a lot, especially James and Kevin for the soybean paper, soybean study, and this group of people who helped with the Forsythia sample, and also collaborators, especially Gary for the samples, and Norman's group, and also Callum for the sequence analysis, and also Department of Energy, BER for funding. Thank you for your attention.
That's fantastic, Mowei. Thank you very much for the grand tour of plant proteomics. Clearly lots of opportunity there, a lot of places where people need to dig in and sort out some of these great questions. We do have a couple questions in the chat, and if folks want to ask a question live, they can just raise their hand and that'll be fine as well. Conrad Winkles has a question. He says, nice talk. Can you elaborate? Oh, wait. I lost my chat.
Oh, sorry.
Can you elaborate a little more on the methods for lysis, prefractionation, et cetera? Anything specific for working with plants?
Yeah, so I probably can show. So it's the plant is a little challenging because it has if you do like a regular type of the whole cell lysis, you get mostly Rubisco protein in your sample. So because it's a very high abundant protein. So for the study I showed at the end, we were doing some very specific cell wall protein prep. So there are a lot of known biochemistry papers, and it really depends on what you are going after. So you should look up those protocols that are specific to like tissue types, or like membrane protein versus soluble protein to really get what you want. Otherwise, you like serum proteomics in humans, like if you don't do some kind of fractionation or depletion, you will see the most abundant proteins, which are likely Rubisco if you're doing whole tissue analysis.
Great, thanks. Kil Wong has a question. He says, great work. I have elementary questions. How complete are the plant protein databases, and how often do you expect to see amino acid substitution in plants?
That's a good question. I'm not a plant biologist, so I cannot, just don't quote on me, but I think it takes, my understanding that it takes a lot of work to really validate and sequence the plant genome. So some of them, like even the published genomes, they're not very well annotated, and there may be some sequencing errors because they're like duplicates of genes in plants. But the substitution, and in general, I do think there seems to be more like close homologs in plants, but I'm not sure. So there may be some sequence variants or homologs because of the larger size of the genome, especially in hybrid plants, because you basically kind of increase the size of the plant by the hybridization, and then you can have basically 2 copies of very similar sequences of protein. So I think that's maybe a little more common in plants.
Great, thanks. I have a question from Kinwing Liu. So it's great work. I'm particularly interested in the way you use top-pick to locate unknown PTMs. Apart from sequence coverage and cross-validation by using bottom-up, what other methods would you suggest to validate those unexpected mass shifts?
So some of them, there are some specific groups that has interesting chemical properties, like they have like specific neutral loss or something that can be really helpful in validating the assignment. And also there are some like classical like chemistry or biochemical methods that are specific for some PTMs that can help you validate. But it really depends on what PTM you're going after.
Great. And it looks like a final question from Julian Whiteledge. Fatty isolation increases hydrophobicity. As you mentioned, have you noted increase in retention time in the LC?
That's also a very interesting question. So we didn't look into that because for that specific protein I detected, it seems like it's almost exclusively modified with the lipids. So we don't have a reference to compare the unmodified peptides or proteoform to see if there's a LC shift. But I imagine it should shift the retention time.
Great. And a question from Jenny Broadbelt. How much optimization did you need to do for the matrix used for the MALDI of intact proteins?
Yeah, that's Kevin was, I don't know if he's on the line, but it does require a lot of optimization. And also, like the washes are important to desalt the samples and also not washing off too many things. And even like if you do imaging with different modalities like nanodesi or MALDI, they tend to give different coverages. I think the extraction step is very important, and there are many variables we don't fully understand yet on what protein get it preferentially extracted by which. I think there's a large space to explore. But the coverage we saw in this study was not overlapping that well with our LCMS data, which is a little surprising because we've done the same analysis for some human tissues in the past or mammalian tissues in the past, and the overlap was better. So I think there may be some special challenges we don't fully understand for plant samples yet.
Great, thank you very much. Any final questions? Okay, if not, I'd like to thank Mowei again. That was really fascinating work, fantastic stuff. And it's interesting to see the plant universe. Oh, wait, we do have a question here. Or is that a clap? Never mind, just a clap. Oh, okay. So yes, thanks again. We appreciate it, Mowei, and we hope everyone has a great day, and we'll be in touch soon. Thank you very much again. Bye-bye.
Thank you. Bye.